Hi everyone, my name is Lena.
I'm Ian.
And we are two of your UTAs, but also we run Tilt.
So we're going to have Tilt tutoring on Sunday evenings from 5 to 9. On Sundays, we're going to also have a Zoom link, so you guys don't actually have to come to the Tilt building if you guys want to stay at home. But we'll both be there that night, again, 5 to 9 Sundays. And then we're going to have another session on Tuesdays. That's going to be 5 to 9 as well with just Elena. There's going to be no online option on that one, so you'll have to come in Tilt. And then I will be tutoring on Wednesdays from 6 to 9. And this is all happening in the Tilt building in the Great Hall. You guys can go online and actually sign up. The schedule's on there. All of that will be on the Tilt website, so you guys can check that out. Yeah, we're here to help you guys crush BMS 300. Yeah, you guys have any questions? What's up?
What are you two doing?
Okay, it's me Sunday nights, 5 to 9. Tuesday nights, 5 to 9. Wednesday nights, 6 to 9. Yep, all in the Tilt building, Great Hall. Yeah.
Is there like a required amount of time that you need to be there?
No, you guys can stop by, ask a few questions, head out. You don't need to be there the full 4 hours. Yeah. Just a drop in.
And come with questions. I mean,
For sure, come with
yeah.
questions. Anything else? Awesome. All right, hope to see you guys there.
Okay.
Thank you, Warren.
Sure. Glad to do it. I really encourage you to go to these Tilt meetings. The folks over there really would like to help you learn. They're there to help you out, and they're very friendly. And we've been doing this for years, and a number of students have moved through there. So I encourage you to do that. Okay, so where we left off last time is we didn't quite get to the secondary structure of proteins. So we have to begin this idea of secondary structure of proteins today, and we're going to see that when we talk about secondary structure of protein, the big idea you need to hold on to is secondary structure of proteins results entirely from hydrogen bonding. We don't need to have any R groups invoked in this story of secondary structure. R groups will be invoked when we start talking about the tertiary structure of proteins. But yesterday we talked about the primary structure of proteins, meaning it was the amino acid sequence. Now we're going to see how the interaction between the amine hydrogens and the double bonded oxygens and the carbonyl oxygen, those charge-charge interactions can now begin to form the shape of the protein. And the beginnings of the shape of the protein are going to be these two big categories, one being alpha helix, the other beta pleated sheet. The alpha helix is going to be hydrogen bonding within the backbone of the molecule. It's within the molecule itself. Whereas the beta pleated sheet is bonding between two adjacent strands. And we'll talk about how these are organized. But the major takeaway from all of this is going to be secondary structure is entirely due to hydrogen bonding. Then we'll begin talking about the tertiary structure of the protein, the three-dimensional structure of the protein. And it's certainly true that a part of this three-dimensional shape of the protein derives from a combination of alpha helical structure and beta pleated sheet structure. But more important in the tertiary structure of the protein is going to be the R groups. We can describe secondary structure without invoking the R groups at all. But when we get to tertiary structure, now we have to begin thinking about those R groups and the amino acids. We have to begin thinking about these R groups as to whether or not they're hydrophobic or hydrophilic. As the name implies, if the R groups are hydrophilic, they're going to be pointing out toward the water. If they're hydrophobic, they're going to be pointing inward toward one another. The way I like to think about it, and it's not probably quite accurate, but I like to think about it as though the hydrophobic R groups are going to be hiding from the water, and the hydrophilic R groups are going to be pointing out, interacting with the water. And because of the amino acid sequence and the position of the R groups, we can now begin to invoke the three-dimensional shape of the protein. It's really hard to overemphasize the importance of the three-dimensional shape of the protein. The three-dimensional shape of the protein is going to determine its function. It doesn't matter whether it's a transmembrane protein receptors in your brain, whether it's an antibody, it's going to be an antibody binding to an antigen, whether it's going to be something we'll talk about toward the end today, where we're talking about the binding of the COVID spike protein to the receptor in one of your cells. All that has to do with the three-dimensional shapes. And so we're going to have to think a little bit about where this three-dimensional shape comes from. And we're going to see that it's a combination of the secondary structure and the orientation of the R groups with respect to water. And I hope there's time. I didn't get to it in the first section. But then what I want to do is think about how we're going to take these proteins and have them traverse the phospholipid bilayer. Remember last time we said blithely we could put these proteins across a membrane and we had them shaped like a hot dog bun. Well, today, if there's time, what we'll see is that whenever we pull a protein, whenever we generate a protein and put it across the phospholipid bilayer, in eukaryotic animals at least, it's organized as an alpha helix. You can imagine it going across the phospholipid bilayer one time. I mentioned yesterday, no, I didn't get to it in here. We'll see today that for each step of an amino acid in an alpha helix, we step up by 0.15 nanometers. And you may remember, I said yesterday, the width of the phospholipid bilayer is about 6 to 7 nanometers. So 0.15, it's going to take about 40 to go across that phospholipid bilayer. And we'll see how they're organized to go across the phospholipid bilayer, how they can then go back across the membrane numerous times and form a channel. The simplest channel I know of is 4 transmembrane regions. But many of these channels contain as many as 24 transmembrane regions, some a few more than that. But let's start here with the idea of the alpha helix. And we'll label this as secondary structure. As an alpha helix. The helix tells you it's a kind of corkscrew. And the helix I'm going to show you here looks a little like this. Now, this is a proper protein as we described yesterday. It has an amino terminus down here. Someplace up here we can put the carboxy terminus. But from yesterday's discussion, you'll recognize that this string of amino acids is going to be the protein. So we can now put a series of amino acids in here, and I'm going to put the amino acids in a circle. So each one of these circles is meant to be, each circle is meant to be an amino acid. Now, if we look at these amino acids, I'll remind you from what we talked about yesterday. When we have an amino acid, we have a nitrogen, a hydrogen, a carbon with an R group, and we're going to ignore this R group for now, and then a C double bonded O, OH. Just as we saw in water, this nitrogen is going to be hogging the electrons. Because it's hogging the electrons, this nitrogen, these two nitrogens will have positive charges on them. Again, this double bonded oxygen standing out here is going to have a slight negative charge on it. Now we're going to use those charges now to hold this structure into a stable state. And we'll see that this hydrogen on that amine for this number 1 amino acid is going to interact with a double bonded oxygen up here. And there's a pattern here. It's a constant pattern called I+4. The reason they use this nomenclature is we can choose a number along that chain of amino acids and call it I, and we'll know that the interaction is going to be between that amino acid plus 4. So if we do the simple one, where we say I is 1 plus 4, that means number 1 is going to be interacting with 1, 2, 3, 4, 5. And there's going to be a double bonded oxygen up here that's negatively charged. And this positively charged hydrogen now is going to interact with this double bonded oxygen. There's going to be a double bonded oxygen on this one that will be interacting with some amino acid I plus 4 downstream. So each one of these amino acids is interacting with amino acids on either side of it in this I plus 4 arrangement because of the hydrogen positive charge and the negative charge in the oxygen. Those charges, the positive charge and the negative charge now, are holding this molecule together. And the reason we can hold the molecule together in this fashion is that we can twist these bonds. They're mobile. So we can twist around this central axis. And by twisting around the central axis, we can now twist these amino acids into this spiral staircase. I think it's useful to think about these alpha helices as spiral staircases. We're all familiar with spiral staircases. And if you can picture yourself on a spiral staircase, you can imagine you start up a spiral staircase and you're facing this wall. And then you take one step and another step and another step. And each time you take a step, you turn slightly. Typically, for a spiral staircase, most of the ones I've seen, it takes 12 or 13 steps to go from facing this wall up the spiral staircase until you're facing that wall again. We can think about this in the same way. We can think about each one of these amino acids as being a step. We can think about the step, the rise, if you will, on the step, being 0.15 nanometers. Where that analogy begins to fall apart is that these are giant turns. Each one of these turns, each amino acid, turns by 100 degrees. From your geometry, you know it takes 360 degrees to go full circle. So that's telling us there will be 3.6 amino acids to go around the circle. So if we use the spiral staircase analogy, each time we step up by 0.15 nanometers, but we don't turn a small amount, we turn like 90 degrees plus a little more. Take the next step, we turn 90 degrees plus a little more. Take the next step, turn 90 degrees a little more, and 3.6 steps, 3.6 amino acids takes us around a full circle. And then they're hydrogen bonded in the I+4 arrangement. That's how we get this spiral staircase. And this is a reasonably stable structure. It's stable because the hydrogen bonding within the molecule is holding it into the structure. Now, a couple of details here. Again, I'll never ask you this kind of thing. But we don't need R groups for this. R groups are not required. But having said that, there are some amino acids that are more likely to form alpha helices than others. And if you take a biochemistry course, you'll probably see this referred to as malic. And if you take a biochemistry course, you'll memorize the single-letter amino acids. Well, this is methionine, alanine, leucine. This is glutamic acid. This is lysine. Those are the most common that form the alpha helix. You're not restricted to these, but they're the ones that are most common. But again, I don't like to emphasize that too much because this implies there's an R group arrangement because we're talking about the different amino acids being differentiated from one another by the R groups. We don't need them for this. This is all internal hydrogen bonding. And that internal hydrogen bonding reaches a stable state by having this arrangement between the positively charged hydrogens and the oxygens I+4 away to throw this molecule into its spiral staircase. This can be reasonably stable in a hydrophobic environment. If you put it in water, it's not quite so clear because these hydrogen bonds then can sometimes switch around and interact with water, and it kind of disrupts the structure. But if we put it in the right environment, if we hide it in the inside of a protein or hide it in the phospholipid bilayer, then it's pretty stable. So any questions on that? Yeah?
Can you clarify exactly what the I+4 means? Like, so is it giving you, like, how many hydrogen bonds?
So what are we talking about with the I+4? That's really the question.
Yeah.
The I+4 is a kind of shorthand. And it's telling us which amino acids are interacting with which other amino acids. Which amino acids are interacting with one another through the hydrogen bonding. If you take two amino acids that are right next to one another, as we drew out yesterday, they're too close to hydrogen bond. They can't twist around that sharp. But we can get twisting. We can get enough twisting so that this number 1 amino acid can interact with number 5, I+4. Number 2 can interact with number 6, I+4. Number 3 can interact with 7, I+4. Now, to make things even weirder, I was reading yesterday that there are some proteins that will actually, some alpha helices that are I+3. They're rare, but they have a few of them. And there's some that are I+5. And so you have either a tighter twist or a broader twist. And that has to do with how fluid we can twist around these angles here. Does that help?
Yeah.
Okay. There's one in the back, and I'll come to you. Yes?
That answered it.
Okay.
So, as you're talking about the structures, a secondary structure can also be 3D, correct?
The question here is, can secondary structure be 3D? Yes, there is some certain level of 3D structure to this. And the next thing we're going to go to is a beta strand or beta pleated sheet. That also has some three-dimensional structure. And you're right in that we're going to mix some alpha helical regions and some beta pleated sheet, and that begins to get a three-dimensional structure. But none of that requires R groups. To get proper three-dimensional folding of the protein requires the R groups and their interaction with one another and their interaction with water. There and there. Let me take this one first.
Okay, does the amine group always, like, with the carboxyl, like, with the I+4, does the bottom one, is it always the amine group that's in the bottom and the carboxyl in the top one?
So, as far as I know, and again, I'm pretty sure this is right, this is the hydrogen, this is the double bonded oxygen. Then this will have an oxygen on it, and it will bind hydrogen bond with oxygen someplace down below the floor in this alpha helix. But that's the pattern. Everybody has an oxygen on them, everybody has an amine on them, and they interact in the I+4 to stabilize that structure.
Okay, but the oxygen's never, like, above?
As far as I know, the only way we can do this is this hydrogen has to interact with this oxygen. And that's determined by the fact that we have this fixed angle around which we can twist things. And so this carbonyl oxygen is going to interact with somebody up here in this direction, and this hydrogen is going to interact with somebody in that direction. But it will be the I+4 organization. There was one here, and then I'll come back to you.
When we see like an alpha helix on like an exam question or something like that, will it always be oriented with like
Okay, so these exam questions, what's going to be on an exam? I hate these questions, but I'll address it. I'm never going to ask you to recapitulate this. I'm never going to ask you about the I+4. What I'm going to ask you about is hydrogen bonding. Hydrogen bonding within the molecule, why this forms an alpha helix and not a beta pleated sheet. I want you to understand it's the hydrogen bonding, but I need to tell the story about hydrogen bonding, so I need all these characters to tell it. I'll not ask you about the characters, I'll ask you about the plot. Okay? Yes? Yeah, yes, sorry.
Okay, so you're saying, like, for example, you have like amino acid 2, and then it pairs with amino acid 6 because of that I+4.
Say that again, I missed it.
So the amino acid 2 pairs with the amino acid 6 because of the I+4?
That's right. So two will go to 6.
Yeah, so is that
So if you-
when it makes a full circle from those binding characters?
Almost. You're right.
Okay.
It's almost the 4, but it's going to be a little more, right? Because the full 360 is 3.6. And now you're going 2 to 4, so that's 4 in between. So you go slightly more than 360 degrees to form that hydrogen bond. But they will form.
Okay.
But you're right. I mean, these are similar, right? We go 360 degrees, we have 4. If it were simple, then every 3.6 would be hydrogen bonding, but it doesn't work that way. And I think it's just simply because of the size of the amino acids, the size of these bonds. Anything else? Okay. Let me go to the other panel. And on this panel, we're going to talk about the other kind of secondary structure. We're going to talk about beta pleated sheet as secondary structure. the emphasis on the alpha helix is that that's hydrogen bonding within the backbone of the molecule. When we're talking about beta sheets or beta strands, we're talking about hydrogen bonding between adjacent strands of the protein. So I'm going to draw this out as best I can, then I'll show you a pretty picture. But we're going to say that this is the amino terminus of the molecule. Actually, let's put it up here. And then we're going to talk about the position of the hydrogens in the R groups. And you can see from the way I'm drawing this why it gets its name as a pleated sheet or a pleated strand. This looks like a series of pleats in clothing. But we can also look at each of these points, these vertices, and say this is R, O, H, R, O, H, R, O, H. Make this an R. And then I'm going to extend it out here and not put any structure in here. And this is going to be an amino acid sequence. Probably rich in prolines and probably rich in glycines that make a kink. So we can twist this around so that now we have the same pattern. Again, here's an R. Now this hydrogen is going to have a charge on it. This is the hydrogen that belongs to the amine. And it's going to pair here with that double bonded oxygen, this carbonyl oxygen. It's negatively charged. Here's an O. This is going to be a positively charged hydrogen. This is another R. This is hydrogen. This is going to be a double bonded oxygen, so it's negatively charged. This oxygen is going to bond to hydrogen, this R to an R, hydrogen to an oxygen. Again, this is negatively charged, this is positively charged, this O to hydrogen. So now what we're doing is we're using the hydrogen bonding. We're using the hydrogen bonding to take these amino acids, and I'll leave you to sort out where all these amino acids are. We're using these hydrogen bonds to hold the two strands together. So now we have the strand sort of running out in space for long distance, loops back on itself, and now it's held together firmly by these hydrogen bonds. This is the bonding, the secondary structure resulting from hydrogen bonding called the beta pleated sheet. There's some nomenclature in here, and I don't want to get too deep into it, but I think it's an important idea if you can see it. This is the amino end of the terminal, or amino terminal of the molecule. So now we're going amino terminal. If we go all the way around here, if this were the end, this would be the carboxy terminal, all right? So we're going amino to carboxy. And then we have amino to carboxy. This arrangement, this has a name. This arrangement is said to be anti-parallel. The reason it's called anti-parallel is that in this direction we're going amino to carboxy. In this direction, we're going to amino carboxy. These are arranged in opposite directions. Even though it looks like they're headed in the same direction, they're said to be anti-parallel because of this. Because we go amino to carboxy, amino to carboxy. That's the anti-parallel. We can imagine, though, that this protein, someplace out here, has some alpha helix. And then it comes back here and it does this. And again, there will be hydrogens up here. I think this is going to be a hydrogen. And it's going to interact with an oxygen. And so now we're going to be holding these together, these two strands. But this organization is said to be parallel. It's parallel because this is amino to carboxy, and this is amino to carboxy. So they're said to be parallel. Let me show you a pretty picture of this. Maybe we can help solidify some of those ideas. This first image is a stick diagram of what I just tried to show on the board. Much prettier, but still same ideas. So this is the alpha helix. And if you study this for a little bit, and it's in the ebook, if you study this for a little bit, you'll see here there's a nitrogen, there's a carbonyl carbon, so there's the peptide bond. Follow it through, here's the carbon with the R group, here's the nitrogen, here's the peptide bond. So that's one amino acid. And if you walk your way through this and you count, you'll find that if you make this the number 1 amino acid, this is going to form a hydrogen bond. Well, let's do it here. This is going to form a hydrogen bond between this hydrogen on the nitrogen and this carbonyl oxygen, number 1 to number 5. Number 5 is then going to interact with number 9. But each one of these amino acids is going to have a mate someplace along this line with which they're going to form hydrogen bonds in the I+4 arrangement. And as the question that referred to earlier, as we go up, this is a hydrogen to oxygen, but this oxygen is going to hydrogen bond with somebody down farther on the helix. But the main idea here, beyond all those details of what each one of them is doing, the main idea is we now have generated a stable structure of a protein, the secondary stable structure, which results from the hydrogen bonding within the backbone of that molecule. So the hydrogen bonding between nitrogens, between the hydrogens which are positively charged because the nitrogens have stolen the electrons, and the oxygens which are negatively charged. We can hold that molecule in a stable state in this spiral helix. It's a major idea of how we're going to make proteins. And it's all done by the hydrogen bonding. We don't have to invoke the R groups at all in this. We don't care about R groups yet. And we don't care about R groups even when we're talking about this secondary structure over here of the beta sheet or the beta strand. Again, here's the amino end. Come around, someplace down here there's a kink. This is toward the carboxy end. This will be antiparallel. And note how these guys lock up very nicely. These hydrogens, these oxygens are pretty close to one another. So that's a fairly strong hydrogen bond. They're being held together fairly firmly. But if we loop around the other way, and we run this parallel arrangement where this is the amino terminus, and this is toward the amino terminus, this is toward the amino terminus, this is parallel, these bonds are not quite so tightly close together. So this bonding is not quite so strong. But nonetheless, we can still hold these beta-pleated sheets together, one to the next. So at this point, we have two kinds of secondary structure, one of which is the alpha helix, and it's bonded hydrogen bonding, making that spiral staircase. But now we have these great long proteins that'll have beta sheets adjacent to one another, and those then can hydrogen bond with one another and hold the two strands together. Now, before we get into tertiary structure, you're absolutely right when you look at this, that you're beginning to see a three-dimensional shape of the protein. We're beginning to see the three-dimensional shape of the protein simply dependent upon the hydrogen bonding between strands within the alpha helices. But there's an additional level of structure. An additional level of structure, and I'll put this in the upper panel. The additional level of structure is said to be the tertiary structure. Now, in some cases, you'll read about quaternary structure, and that's where multiple subunits come together. But for our purposes here, we're just going to go as far as the tertiary structure. And the major idea to come out of the tertiary structure is that the tertiary structure is going to be influenced tremendously by the characteristics of the R group. So what I want to do here is say that hydrophilic and hydrophobic R groups. I won't say determine, I'll just say influence. The 3D, which you can read as tertiary structure of the protein. There was a question out here. Yes, you.
On the beta pleated sheet, the corkscrew end, is that the carboxy end?
Oh, I see what you're asking. So your question is about this. Your question is about this alpha helix that I've put in the middle of the protein. Yeah. Well, if I make this the amino end, we're walking this way, we're walking toward the carboxy end. This end of the alpha helix will be the amino end, right? And this end will be the carboxy end, but it will continue on until we get to the carboxy end someplace out here. Yes?
So for the beta pleated sheet, you said it was between different strands. Are the strands the part of the same amino acid chain?
So the question is, are these two strands, we have adjacent strands, are they all part of the same amino acid chain? Yes, they're all part of the same primary structure.
Okay, so the difference between the alpha helix and the beta pleated sheet is the alpha helix bonds to amino acids closer to it than the beta pleated sheet?
Yes, they will be more, and they'll bind to one another, closer to one another, and they're also binding in such a way they're twisting around those other bonds in the amino acid. Here you don't get the twist. They're laid out as a zigzag pattern, and they're interacting chain to chain. Yes?
So is there a way to know which way it's going to be bonding, or do you just like you get an example and really like have to know how it's working?
It's a great question. Is there any way of knowing this a priori? And the answer is probably not originally. That we don't know exactly how this is going to end up, and most of this structure comes out of doing X-ray diffraction and by looking at each individual protein. Now, having said that, there was a paper that just came out a few days ago. It was in Nature. And there is an artificial intelligence group in England called DeepMind. It's part of Google. And they have applied supercomputers to trying to understand how proteins fold. And they've gotten pretty good at taking the simple amino acid sequence and predicting the three-dimensional shape. And so what they did is they took some proteins they knew from X-ray diffraction. What's the three-dimensional shape? And they then gave the computer the amino acid sequence and how's this thing going to fold? And it predicted it remarkably accurately. So from that, we now think we can take the amino acid sequence, which we can get from the RNA and DNA, take the amino acid sequence, and once you simply know that the series of amino acids in the string, we can predict from these computer models how they're going to fold in a three-dimensional shape. That's going to help us tremendously in generating drugs and generating binding pockets and getting proteins to bind to one another. So it's an enormous step forward, and it's all done through artificial intelligence in the computer. But you're right. Up until this, people have been trying to predict this for the last 50 years, and they just didn't have the computer power to take in all of the variables. But now they're pretty good at it, apparently. How it gets done is beyond me, but it's been done. Anything? Yes?
Can you draw the ROHs on the parallel string?
Oh, man, I'm not sure I can. Tell you what, I'm going to show you the slide. The ROHs in the antiparallel strand are easy because it's the mantra of ROH, ROH. Now, if I can find my pointer. So the right-hand panel here is going to be the parallel arrangement. So if we walk through that, on this strand, here's RHO, RHO. Here, the double-bonded oxygen, here's the hydrogen. R is out in space. Here's the hydrogen, double-bonded oxygen, R is in space. O and the H. So if you want to memorize that, you're welcome to it. All I'm going to ask you to know about is that within these strands, between the strands, we have the hydrogen bonding holding them fast together. And by combining alpha helical regions and beta pleated sheet regions, we begin to see the three-dimensional shape of the protein. In fact, it's maybe worth looking at a couple of slides here with that respect. So when you read a paper, you're going to see protein structures described. And the shorthand for those protein structures is to show a ribbon like this. And then the arrow points toward the carboxy terminal. And in this case, these are all set up anti-parallel. Here's toward the carboxy terminal, toward the carboxy terminal. Now, in some cases, I think the next protein shows some of these regions which will be parallel. Now, this is, again, beginning to show the three-dimensional shape of the protein. We don't have the full three-dimensional shape at this point. But what we have in magenta here are regions of the alpha helix. This is as though you've taken a ribbon and taken your scissors and pulled it along the ribbon, and it throws it into that alpha helix. So we have regions that are bound one to another internally with the alpha helix. Then there'll be other regions in here that are beta pleated sheet or beta strands, some of which are running parallel, some of which are running anti-parallel. That begins to demonstrate the three-dimensional shape of the protein. But we can't get a full meaning of the three-dimensional shape of the protein until we invoke the R groups. It's the R groups and their interaction with one another and the interaction with water, which will determine the three-dimensional shape of the protein. And again, it's really hard to overemphasize the importance of the three-dimensional shape of the protein, because it doesn't matter whether we're talking about neurotransmitter receptors in your brain or whether we're talking about antibodies binding to antigen. That all has to do with the three-dimensional shape of the protein and its interaction with its three-dimensional target. If you put your phone away, I'd appreciate it. Thank you. Yes?
So as far as like the portion of the protein that is parallel or anti-parallel, is one of those a more stable form than the other?
I'm not sure that one's more stable than the other, but that anti-parallel arrangement, because those hydrogen bonds are closer, I think those hydrogen bonds are going to be stronger. But both of them are reasonably stable in that you have a low order state of hydrogen bonding holding these things together. But to really get the three-dimensional shape of the protein, we have to invoke the R groups. So any other questions before we go on to tertiary structure? Okay. Now I'm going to show this as a kind of ribbon. And this is a gross simplification. But I think it points out some important ideas. I'm going to show the protein as a ribbon, and I'm going to imagine on that ribbon that there are R groups. And so we can draw one of these in blue. So this is meant to be the backbone of the protein. This is a really oversimplified protein. And then that protein is made of amino acids, and so there will be R groups on here. And interspersed among those R groups will be some others. Now remember, R groups come in three varieties. One is they can be uncharged. Examples of these will be leucine and isoleucine and valine and some others. Don't memorize those names, I don't care. But know that there are a subset of amino acids that are uncharged. They're neither polar nor charged. There'll be another set of amino acids which are polar. Among the polar amino acids will be things like serine, which has hydroxyl, and threonine. Both of these have hydroxyls. Another one is tyrosine, which has a hydroxyl. Those are all polar. The third group are the ones that are properly charged. And among the charged ones will be things like glycine and glutamate and arginine, I think arginine, and aspartate. Now, of course, because they're polar or charged, that tells us they're hydrophilic. These guys that are uncharged, these are hydrophobic. Now, in this simplistic view of the straight ribbon of the protein, the amino acids, when this thing is exposed to water, you can imagine how it's going to reorient itself. It's going to reorient itself in such a way that these proteins will fold so that the hydrophilic R groups are pointing outward to water, and the hydrophobic R groups are pointing inward and hiding away from the water itself. So this is hydrophobic. And these blue ones are hydrophilic. Now, having said that, this is a poor representation because it doesn't really show the three-dimensional shape. I'm trying to show this as though it's a squiggle. But this will have proper three-dimensional shape. Now, an example I want to use for three-dimensional shape and how it will function, and I'll put this over on the right-hand panel, is a molecule we're going to run into later. We're going to run into this when we talk about cell biology. We're going to run into this molecule when we talk about muscle. And the molecule I want to spend a moment on here, and thinking about its three-dimensional shape, is one called myosin. Myosin, we'll see later, myosin is something known as an ATPase. It converts ATP into adenosine diphosphate plus inorganic phosphate. It's an enzyme. And it has a remarkable three-dimensional shape, but we can draw it more or less in 2 dimensions. It has this tail on it, and it has the thing called a head. And this head, this divot in the head, is the ATP binding site. If you think about it in 3 dimensions, you can describe it as an ATP binding pocket. The 2-dimensional shape doesn't really do it justice. It's a proper 3-dimensional structure. And this proper 3-dimensional structure, let me make it a little bigger over here. This proper 3-dimensional structure is, of course, composed of amino acids. And some of these amino acids will be polar. And tucked away, some of these amino acids will be nonpolar or hydrophobic. Some will be charged. But this structure now is folded in such a way that it has a particular shape. It's a particular size and a particular shape that's dependent upon the charge-charge interactions in the protein, the charge-charge interactions between the R groups in the water. This gives it a certain shape so that precisely into this structure fits adenosine triphosphate. This is the binding pocket for ATP. Adenosine triphosphate is a pretty good-sized molecule. It fits into this site. It tucks in very closely. And it tucks in so closely that the charge-charge interactions between ATP and the amino acids in the binding pocket lower the energy of activation so that we can convert adenosine triphosphate into adenosine diphosphate plus a molecule inorganic phosphate. We're just clipping a phosphate off. In this particular molecule, when we take ATP and convert it to ADP plus inorganic phosphate, that can reside in this binding pocket for a long time. It's stable. But what will happen, and we'll see this in much more detail later in the course, that this myosin head can bind to an actin. This is an actin. And the binding of the actin to the myosin changes the structure in this 3-dimensional binding pocket, and the ADP and PI leave. But when the ADP and PI leave, this myosin head goes from a more or less upright position to being folded over. And as it's folded over, because it's attached to the actin, it's tugging on the actin. That's how your muscles are shortening. Well, all I want to point out today, and we'll get into this detail much later, but what I want to point out today is this myosin head has a binding pocket, and part of its three-dimensional organization, dependent upon its amino acid sequence, allows for the ATP to fit snugly into that site. So snugly does it fit into that site that charge of this molecule. The molecule that we're looking at is the spike protein for the coronavirus. And this first iteration of the structure of the spike protein is its amino acid sequence. This is the primary structure. And it's showing here the amino acids between 1 and 1273. This is the amino terminus. This is the carboxy terminus. Each of those amino acids is carefully known. It's accurately known. We know the order in which those amino acids exist. And different regions of the protein in its amino acid sequence and its primary sequence do different things. So this region here, between 319 and 541, again, we're numbering the amino acids from the amino terminus to the carboxy terminus. The ones in this region are called the receptor binding domain. This is the region of the spike protein that binds to receptors on your cells, called ACE2. The way this molecule gets into your cell is that the receptor binding protein region on the spike protein binds to one of your cells, a receptor on your cell. We know how this binds, and that's shown in the next slide. In gray, we're showing all this alpha helical region. And in gray, we're showing the alpha helical regions of the amino acids in something called the ACE2 receptor. These are on all your epithelial cells in your respiratory tract. And then here's the amino acid sequence of the receptor binding domain. And they're showing each one of the amino acids in the ACE2 receptor and which amino acids they bind to in the receptor binding domain. Now, this is a detailed figure showing some of the particular mutations that have occurred. The mutations that they were worried about when I made this slide were up here at N501Y, where this amino acid had been changed to a tyrosine. And there are a few others in here that were changed. But what we think is happening with the delta version, if we go back, is we think that what's going on in the delta version, and this is a paper that just came out a couple days ago, is that in this region around 683, there's a junction here between something known as S1 and S2. The junction between S1 and S2 is susceptible to proteolytic enzymes. And so what happens when this binds to your cell is a proteolytic enzyme will cleave the spike protein at that point. And when it's cleaved at that point, that leaves part of the spike protein with a hydrophobic series of amino acids, which can now bind with the phospholipid bilayer in your cell and burrow into the cell. What we think is happening in the delta protein, the delta mutation, is that they've changed a proline at position 683 into an arginine. And the more arginines you have in this site, the more susceptible they are to the proteolytic digestion, so it's more likely that you'll have an active virus that can infect the cell. We think that's why it's so more infective. Let me show you this last picture before you go. this is the three-dimensional shape of the spike protein. And if you look like here, out here, this is the receptor binding domain. That's just a little piece of it. And down in here is S1, S2. That's where we split it into two parts. So we've gone here in these three figures to show the primary amino acid sequence, 1 to 1273. Then we showed how the alpha helical regions will bind to the ACE2 receptor. And now this is the overall three-dimensional shape of the protein. And the overall three-dimensional shape of the protein has these number of different regions which will do different things. This is the part that binds to the ACE2 receptor. This is